Good afternoon, everyone. This will probably be the highlight of the day, hopefully. Some really brainy young researchers are going to take you on a special journey into cannabinoid science. We've got 10 speakers. They've each got about 5 minutes. And we'll hold questions until the end. So a bit like cannabinoid scientific speed dating or something like that. I'm Iain McGregor from the University of Sydney, and I'm joined here by
Veronica Borrett. So I'm from the Australian Research Council Hub for Medicinal Agriculture, and we're really pleased that the organisers developed this session, because we all know that there are huge gaps in the understanding right across the value chain for medicinal cannabis. And so, we need a lot of research, and so today we're pleased to be able to hear from our 10 speakers to give a snapshot of the research that they're involved in, or I guess a taster of the research they're involved in. So with that, our first speaker is Laura Steele, and she's a PhD student at the ARC MedAG Hub, so she works with us, and she's speaking about the investigation of flowering time in cannabis.
Thank you for that beautiful introduction. I don't think I really need to introduce myself, but yes, I'm Laura, and I'm excited to share my research on flowering time in cannabis with you today. So when we talk about flowering time and its impact on plant morphology in cannabis in particular, really what we're saying is that early flowering time has a notable impact on plant architecture, resulting in shorter plants with fewer branches, while later flowering is known to result in taller plants with a higher degree of branching. And this has implications for phytocannabinoid yields, because our early flowering plants, we may be able to fit more of them into a square metre. We have increased harvest frequency, which is preferable from an industry perspective, but conversely, a later flowering plant will support a higher volume of floral biomass. So with that in mind, my research aims to identify genetic factors regulating flowering in cannabis to optimise flowering behaviour to improve phytocannabinoid yields. And to do this, I started by looking at the segregation of flowering time traits in early and late flowering accessions. I conducted a trial where I did a destructive harvest of plants, and I found that there were positive correlations between an increased time to maturity and, unsurprisingly, taller plants with greater biomass. Interestingly, the opposite was true for our early flowering plants, which after some cannabinoid content analysis also demonstrated that there was a negative correlation, that is that an earlier flowering plant was higher in cannabinoids. Interestingly, again, when you look at the dry flower produced by individual plants each day, that is adjusted for time to maturity, we still saw a greater increase in flower in our later flowering accession. So then I decided to cross our early and late flowering lines with the intention of generating what's known as an F1 population. And I grew these out to look at how closely they resembled either or both parents. And we saw an intermediate phenotype, that is to say that they had variation in orders of branching, but height-wise, they tended to favour their taller, later flowering parents, while the flowers themselves, the inflorescences, more closely resembled the early flowering plants. That is to say that they had shorter internodal spaces and resulted in more dense inflorescences. The frequency distribution you're seeing here also indicates that while there was variation, that was to say that flowering time was normally distributed within the population, again, it very strongly favoured that of the early flowering parent. So what I'm currently doing is I'm looking at, with an F2 population, that is plants that resulted from a self-cross of F1 individuals, growing these plants, scoring them in terms of flowering time behaviour, with the end goal of then taking DNA samples from these plants to do something called bulk segregant analysis, which will look at pools of early and late individuals with the aim to integrate that phenotypic data that I've collected with genomic information to ultimately identify the genetic components that are governing these differences in flowering behaviour and subsequently phytocannabinoid yield. And I'm keeping it short and sweet today in the interest of time. So in closing, I would just like to thank the team at Trobe, in particular the MedAG Hub, as well as all of our industry partners. And I think we're doing questions at the end rather than
We
now.
can do questions at the end. But
Sadly, I won't be here at the end, so if we had time, I'd be happy to take any questions.
Yeah.
Thank you.
Any questions? No? Well, you might think them up. Sorry, is there one there? Great work, yeah.
Oh, there we go, 4 minutes. I was told 4 to 6 minutes.
And just a reminder too, along the way, feel free to post the questions on the Q&A and we can address all of those at the end, but thank you. Yes?
We have a question here.
Yep.
Yeah.
Really great. I love this session. Thank you so much. As a
Thank you.
horticultural research scientist, super excited. My name's Emily Rigby. Could you tell us a little bit about how you grew the plants? Like, were they done hydroponically with supplemental lighting, indoor?
Yeah, absolutely. So they were grown in controlled environment rooms. Essentially, I took cuttings from early and late mother plants, propagated those for sort of 9 to 14 days before transplanting them into a peat, vermiculite and perlite 1:1:1 mix with 1 gram dolomite mine per litre. Apologies if that's very specific. And then they were essentially, in the pilot trial study, not vegged because we wanted to score the flowering behaviour. And so we put them straight into a short day photo period, again, controlled environment conditions under metal halide lighting.
Thank you. Any other questions for Laura? Because she won't be able to stay till the end.
Sorry.
Okay. Thanks very much, Laura.
Thank you.
That's great.
Our next speaker is Dr. Cilla Zhou from the Lambert Initiative at the University of Sydney, who's going to talk about surveys at the Lambert Initiative.
Hi everyone, my name is Cilla and I'm the Science, Communications and Media Manager at the Lambert Initiative. I recently finished my PhD in pharmacology in the preclinical team. Yeah. I am short. And I'm now working to expand community engagement in the clinical team. Today I'd like to talk to you about the CAN-ACT study. So the LI mission is to research and develop novel cannabinoid-based therapeutics, but we are also heavily invested in policy and advocacy on behalf of cannabis users, and this is reflected in the current surveys that we have running. We have the MedFed study, which is an international survey investigating the use of drugs in the treatment of eating disorders, which has seen great success. We also have the Cannabis as Medicine 2022 study, which is a nationwide Australian study conducted every 2 years, and you'll hear more about that with Llew later in this session. And finally, we have the CAN-ACT study, which investigates behaviours and attitudes of cannabis cultivators in the ACT and has only received 195 completions so far. I'll be talking to you about how community engagement has taught us a lot about why we are seeing low engagement numbers with this study. So the ACT decriminalised small cannabis use and possession in 2020, which provides a unique opportunity for us to study cannabis decriminalisation in a local community setting. The study consists of an anonymous survey, collection of participants' homegrown cannabis, analysis of these samples, and delivery of personalised results to the participant. We want to understand choices around cultivation, the medical conditions being treated, the cannabinoid and terpenoid content versus what participants perceive them to be, presence of any contaminants, and finally, community needs, their lived experience, and their feelings around the laws. And here is our study coordinator, Isabel Lavender, who is also doing a PhD with us. The cannabinoid and terpenoid analysis is spearheaded by Dr. Richard Kevin and Rebecca Gordon, who are our analytical chemists. We only require about 0.5 grams of plant material for these analyses, and an additional 4 grams for contaminant analysis. Participants can submit 2 samples, one from each of the 2 plants that they are legally allowed to have. This is an example of what participants will receive in their report. Overall, we're seeing a large variety of high THC to high CBD samples with interesting minor cannabinoid profiles. This is an example of the terpenoid report, and we are hoping again to see a variety of different profiles. It's unsurprising to see high levels of beta-caryophyllene and alpha-pinene, but the dominance of some of the other terpenoids is super interesting. For contaminants, we're working with Chem Centre in Western Australia and testing for several different contaminants. We want to emphasise to participants that it is important to know what you are consuming. Currently, we only have 195 survey responses and 36 samples, but we know there are many more growers out in the ACT. We want to share some preliminary results with you, but we hope that more survey responses will help us get a more accurate view of ACT growers' attitudes and behaviours. Most growers source their plant from seed and convert their plant material to other products, such as tinctures, oils, or butters. 51% of growers are scared of being arrested or charged despite following the laws, and most participants are growing because it's cheaper than prescribed cannabis. Most interestingly, though, I think, is that most growers grow cannabis because they enjoy the process. We've employed a number of recruitment strategies for this study, which you can see here. We spend a lot of time and resources on recruitment, as it's important for surveys to be well-powered with high participant numbers in order to achieve meaningful results. This allows us to use the survey results to shape future clinical trials that will best serve the needs of the community. Because of poor participation numbers, I engaged with members of the community through Reddit forums and came across a number of concerns which deserve to be highlighted. There are 4 main categories. First, there is institutional distrust. Secondly, many growers say that their samples are not ready to be harvested yet, which is fair enough. Thirdly, there is a general fear of criminalisation, which is also reflected in our preliminary survey results, where folks are scared of being arrested despite following the laws. And finally, there are financial implications for growers. We have addressed these concerns, and I'd like to emphasise that participant safety has been a priority throughout the duration of this study. I also believe it is important to gain the trust of participants through engagement with them rather than feeling entitled to their trust. So firstly, we have spent almost a year working with ACT and New South Wales governments to acquire the appropriate permits, and these licences are accessible on the CAN Act website. The survey also doesn't ask for any identifying information, and addresses cannot be linked to the survey. Lastly, our courier has been legally bound to destroy addresses upon collection. We hope that with more participants, we will achieve more meaningful and impactful results. We hope that this study will model what ongoing community testing could look like. We already know that participants have been using their results to make informed choices about their medicinal consumption, and this was featured on ABC News Canberra over the weekend. We hope that we can also help iron out the grey areas in the law for participants and help with the relaxation of cultivation laws to better protect their experiences. We also know that there will be many implications for industry. So for more information about the study, you can visit the CAN Act website, or you can find me after at the networking event, and I'd be happy to chat about the study and our recruitment strategies. Thanks so much.
Thanks very much, Cilla, and we'll hold questions until the end. Next, we welcome John Laws from the University of Adelaide, and John is a PhD student, and he's going to be talking about neurological bioactivity of cannabis prevalent terpenes, implications for medicinal cannabis formulations. Thanks very much, John.
Thank you. Have you ever had a loved one who can no longer remember who you are? Well, I did, and her name was Ruth, also known as Mama. Mama lived 101 years, and at the age of 95, she was sharper than me. Thereafter, she developed dementia-like symptoms and eventually was taken to a nursing care home with Alzheimer's disease. That's not supposed to come up just yet. Alzheimer's disease is the most common form of dementia, and it affects millions of people worldwide, costing healthcare industries millions of dollars annually. Although several approved treatments do exist, they only afford transient symptomatic improvements, are not disease-modifying, and create adverse side effects. And I don't know why that's still happening, but that's besides the point. Maybe I should just speak faster. Well, This underscores the imperative of bios-respecting sustainable plant sources that target or could be targeting neurodegenerative disease. Luckily, nature provides us with Cannabis sativa L, which is a sustainable and efficient plant ally that has been recognized for thousands of years for psychoactive and therapeutic activities. And now with a burgeoning access to medicinal cannabis formulations globally, research has turned to reevaluate cannabis's myriad chemicals for their neuroprotective abilities. Additionally, cannabis produces over 200 aromatic terpenes. Terpenes are responsible for the flavor and aroma of the various C-sativa chemovars. Terpenes have the potential to protect neuronal cells from the toxicity caused from amyloid beta. However, there has yet to be comprehensive studies exploring the potential synergies of terpenes and other cannabis phytochemicals to preserve memory. Therefore, the aim of my PhD is to explore the neurological bioactivity of cannabis prevalent terpenes, which may have implications for medicinal cannabis formulations. This will be achieved by characterizing myrcene, metacarpholopine, fredolin, alpha and beta pinene, linalool, alpha-bisabol, neurolidol, citronellol, terpeneol, limonene, terpenoline, and humulene for their neuroprotective abilities. Thus far, in PC12 neuronal cell culture, we have discovered that alpha and beta pinene had significant degree of neuroprotective abilities at preserving cell viability. Also, myrcene, beta-carpholopine, and alpha-bisabol in undifferentiated and differentiated neuronal cells had significant abilities of also protecting neuronal cells against the neurotoxic amyloid beta 1 through 42. Another assay that we use to evaluate the abilities of these terpenes to reduce the aggregation of amyloid beta, which is a pathological hallmark, and its aggregation, accumulation, disposition is one of the leading causes of Alzheimer's disease. We found that myrcene, beta-carpholopine, and alpha-bisabol all had significant abilities at reducing aggregation and fibrillation, and also terpeneol, terpenoline, fredolin, alpha and beta pinene to a notable degree. To gain more of a qualitative perspective of the density and the fibrillation of amyloid beta and how these terpenes could prevent these densities, all terpenes did have some nice abilities at doing this, but the photograph of alpha pinene is the best representation of how you can see how this particular terpene reduced the density and aggregation. So, will cannabis aromatics be agents for memory problematics? Or will full spectrum extracts, such as Jack Hare, high in alpha-bisabol, or Blue Dream, high in alpha-pinene and beta-carpholopine, be agents to preserve memory so people like Mama will be able to remember your name to her last breath? Well, this research has identified myrcene, beta-carpholopine, alpha and beta-pinene, alpha-bisabol, ability to significantly enhance neuronal solubility and reduce aggregation from the neurotoxic amyloid beta protein, thus contributing to a body of knowledge towards potential applications in the emerging area of medicinal cannabis research in Australia and globally. I thank you all for listening. I do apologize about my slides skipping earlier, but thank you.
Thank you, John, and we'll take questions at the end of the session around that one. Our next speaker is Llew Mills from the Discipline of Addiction Medicine, University of Sydney, who's going to talk about the Cannabis as Medicine surveys and their evolution.
Hi, everyone. My name's Llew. I work at the Specialty of Addiction Medicine, University of Sydney. So this is the group of people who are responsible for CAMS, of which Iain is one. So as Hunter said earlier, people have been using cannabis medicinally for 5,000 years. It's only the last 100 that it's been illegal to do so, which has meant that if people wanted to use cannabis medicinally, they had to break the law. In 2016, as you all know, the federal government deregulated medicinal cannabis use. So it's estimated that about 10 to 20% of people with chronic pain are now using, or in palliative care now use medical cannabis. Yet in 2016 we didn't know much about them, and importantly we didn't know a lot about the people who were doing it illegally. So SASB approvals have skyrocketed over the last, since it was deregulated 6 years ago. So the aim of the Cannabis as Medicine surveys, they're biennial surveys, was to examine the consumer experience of medical cannabis use over time. It's anonymous and online, anonymous being important because it meant people could confess to illegal activity, and people had to be Australian and had to have used cannabis for medical purposes in the last year. So we've had 3 iterations so far. We've had fairly robust recruiting, as you can see, and we're on to CAMS 22 now. We're still taking respondents for that survey. So we'll jump right into the results, but I will start with this slide. So the important row here is the bottom one. You can see that in CAMS 16, less than 1%, a lot less than 1% of people were actually using prescribed medical cannabis, which isn't surprising given that it had only been deregulated a few months before. 2 years later, somewhat disappointingly, it was only 2%, about 25 people out of 1,700 responses. Encouragingly though, in CAMS 20, we had about 40%, and it's now looking in CAMS 22 like that's flipped and we've got about 60%, which is fantastic because we can start to look at the differences between prescribed users and illicit users. So, does this take me back? Doesn't matter. Okay, so the fact that we have more people who are getting prescribed use suffuses itself through all these results. So you can see that the age that people first used in CAMS 20, it's increased from CAMS 16, so people are saying that they started using slightly later in life, and the age that they first used regularly was slightly later as well. The proportion of people who had never used any medical cannabis before using medical cannabis increased significantly. And the days that they'd used was about the same. The total cannabis use for medical purposes hasn't changed an awful lot, and the weekly cost was quite high in 2016, dropped a little bit in 2018, and then increased again in 2020. The main route of administration, it's dropped a lot. People are smoking a lot less. They're vaporising and using oral formulations a lot more. Encouragingly, people are now discussing their cannabis use with their medical professionals, which is probably caused by the fact that we now have more people who are using prescribed in our survey, but that is encouraging. The people who say that, the proportion of people who are saying that accessing prescribed medical cannabis is easy and straightforward has increased a lot. Once again, this has to do with there are more people, more people who have had exposure to the legal system. The proportion of people who said that there were barriers to who said that their medical practitioner being unwilling to prescribe or unable to prescribe as a barrier to their using has decreased. But interestingly, the proportion of people who say that the cost of prescribed medical cannabis is a barrier to them has increased, so that's cause for concern. So what's changed? In summary, there are more people who are new to cannabis. In other words, there are people now that the prescribed users are a slightly different bunch from the people who were using illicitly before. There's more people who had never used recreationally before they used medically. There's greater uptake of oral and vaporised routes, which is encouraging because they're safer, obviously. Also, there's the fact that there are more people who are having it prescribed means that they're getting exposure to healthcare professionals when they're doing it, which means that they're not doing it for conditions for which there's very little evidence for its efficacy, like anxiety and a lot of mental health conditions. They're using it more for pain and things like that. So what's the same? Well, there's a similar range of conditions, although the spread has changed a little bit. Most people are using it for mental health, which is a little bit concerning, but more people are using it for pain now and a lot for sleep as well. So this was part of a much longer talk. I'm running out of time. But yes, we're recruiting for CAMS 22 at the moment, so it's very important that we get more people who are responding to this, because as I'm sure you've all discussed and heard discussed, We need data to be able to present to policymakers and bureaucrats in order for them to listen to us. And this survey gives us that sort of data. It tells us the problems people are having, what they're using it to treat, and quite encouragingly the fact that they're talking to their doctors and their doctors are helping them make better decisions about how to use it. Thank you.
Thanks very much, Llew. I just reiterate to this audience that we're keen to get more people in the CAMS 22 survey. I think we're about 1,500 at the moment. So an unabashed plug, please, if you're using medically yourself, to do our survey. If you have friends, colleagues, patients, then please encourage them to do that, because it's been an incredible tool to change policy in Australia. So let's keep that rolling on.
Thanks. Our next speaker is Matthew Nolan, and he's a PhD student at Southern Cross University. And is Matthew with us? Great. Thanks, Matthew. So as I said, he's a PhD student with Southern Cross University, and he's going to be talking about Cannabis sativa, a model species for glandular trichome research. Thanks very much, Matthew.
Hello. As described, yes, I'm working under Tobias Kretschmer and Bronwyn Barkler at Southern Cross University. My PhD is looking at what drives productivity in cannabis glandular trichomes. Okay, so why trichomes? Trichomes are the glandular structures found on cannabis flower and leaf tissue, and is the major site for cannabinoid production. They're made up of 3 cells: the stalk cells, the stipe cells, and the disc cells. We can see the disc cells are the major site for cannabinoid and terpenoid biosynthesis. The stipe cells connect the disc cells to the stalk cells, and the stalk cells connect the disc cells and the rest of the trichome to the leaf tissue, where energy is transported through the stalk cell into the disc cell for the production of cannabinoids and terpenoids, which are stored within the extracellular space above the disc cells within the trichome. In picture C there, we can see quite a pretty picture of a trichome next to each other. In the stalk tissue there, the glowing red is chloroplasts, which are responsible for energy production, and them being not present in the disc cells demonstrates that energy production is not occurring within the site of biosynthesis. It is being transported from the leaf tissue into the disc cell via the stalks. So how have I gone about understanding the productivity of trichomes? I've taken a proteomic approach, and to start off with, to map the proteome of cannabis trichomes, I needed to isolate trichomes in large amounts in highly purified fractions. So using a method I developed using mechanical disruption and various sieves, I was able to isolate trichomes that were, as we can see in figure A here, which has still intact and still contain all the cannabinoids present with the trichome. In figure B, we can see that they've actually been popped. The resin has been removed, and we only have the cells present. And in figure C, we have just the stalks. Picture D is a photo of a falcon tube of popped trichome heads, and this has then been used to generate a swath library, which is a library containing proteins present within cannabis trichomes only. I have used over 10 different cultivars, both traditional landrace cultivars and modern high-yielding cultivars, to generate the library, as well as producing subcellular fractionations of the trichome disc cells using free-flow electrophoresis and sucrose gradients to allow for resolution of low-abundant proteins within the disc cells. So if we look at the first experiment that I've conducted, is looking at cannabis trichome development through the flowering period. I sampled cannabis flower from weeks 3 through to 8 and quantified the 17 major cannabinoids present within the flower material. We can see in figure B and C that CBDA and THCA had a general trend towards accumulation throughout the flowering period. But it's important to note the major precursor metabolite CBGA remained constant throughout the flowering period. The second part of this developmental study looks at the proteome of the disc cells themselves. So trichome disc cells were isolated from each each plant during the flowering development period and subject to proteomic analysis. The PCA plot on the side here, along with over close to 1,600 proteins were isolated within the disc cells themselves against our library, which contains 2,720 proteins from trichomes only. We can see in the PCA plot on the side of the diagram here, so we start off at week 3 and through to week 8, we can see significant protein changes within the proteome of the cannabis disc cells. Further analysis will look to quantify the changes of individual proteins during the flowering period. We have mapped most of the primary and secondary metabolism pathways within the disc cells, and that includes cannabinoid and trichome and terpenoid biosynthesis. So taking a lower resolution, last year Sam Livingston, a PhD student at UBC, produced some seminal microscopy pages detailing the subcellular organisation of trichomes. So we can see in figure A here that they're filled with plastids, which are responsible for the biosynthesis of the cannabinoids and terpenoids within the disc cells of the trichomes. There are also many mitochondria present within these disc cells, which are responsible for energy production to facilitate the biosynthetic pathways. It's important to note that the plastids are very close to the top of the storage cavity for transport along the surface wall there, which is also known as the plasma membrane. The cannabinoids themselves are actually toxic to the disc cells themselves, down the bottom there. So the trichomes have developed quite a sophisticated mechanism to transport and store the cannabinoids in the extracellular space without them refluxing back in and causing the death of the trichome. So to try and understand how this was coming on, using fractionation, subcellular fractionation of the trichome, the disc cells, I was able to break the disc cells up into tiny little vesicles and then subject them to fractionation via high centrifuge sucrose gradients, which separated the different membranes of the organelles within the disc cells themselves. So on the top layer, we have plasma membrane and tonoplast enriched fractions. The second layer has endoplasmic reticulum and Golgi, and the third one, and the pellet, has plastidal and mitochondria. So, for cannabis, this is a great step forward in getting to understand how these biosynthetic factories work, but in plant science as a whole, I'm suggesting that cannabis should become the model species for trichome research. Previously, it has focused on tomato and petunias, but with the large number of trichomes in highly purified fractions that I've been able to generate through the mechanical disruption, it will facilitate much more in-depth research into trichome development and research. Thank you.
Fantastic, thank you, Matthew, that was terrific. Our next speaker is Danielle McCartney from the Lambert Initiative, who's going to talk about a red hot topic featured in Richard Miss Halley's Fireside Chat, the effects of CBD and THC on driving.
Thanks, Iain, and good afternoon, everyone. My name's Danielle, and today I'm going to be speaking about the effects of cannabis and its major cannabinoid constituents, so CBD and THC, on cognitive function and driving performance. Ooh, went too far. How do I go back? There we go. Sorry about that. Okay, so we recently conducted a study looking at the effects of CBD on driving and other sort of, driving-related factors. So as you can see here, our participants came into the lab on 4 separate occasions, and on each occasion they received a different dose of CBD, all the way through to 1,500 milligrams. They then completed a series of assessments, which included a simulated driving task, some cognitive function tests, and we administered some oral fluid roadside drug tests. Now these tests are designed to detect THC, not CBD, but as CBD does have some structural similarities to THC, we thought, well, maybe there's potential for it to also elicit positive tests. So we wanted to have a look at that. Overall, we found that CBD did not affect driving performance, did not affect cognitive function, and did not elicit false positive roadside drug tests to THC, and therefore concluded that CBD can be used safely by drivers.
Then we went on to have a look at the effects of THC, or more specifically, we conducted a series of meta-analyses investigating the effects of THC on various driving-related cognitive skills, shown on the left, I think, hopefully, and on aspects of driving performance on the other side. And as you can see, THC did have a significant detrimental effect on the majority of these outcomes, leading us, unsurprisingly, to conclude that THC can impair driving performance. Once we'd established that THC could impair driving, we were really interested in the duration of THC-induced impairment. So we took all of our data that we'd used in our previous meta-analyses and produced or conducted a series of large meta-regression analyses predicting the duration of THC-induced impairment, and that's sort of what's shown here. So on the y-axis, what you can see is our measure of impairment with more negative values indicating increased impairment, which is a little bit backwards, but bear with me. And then on the x-axis, our time post-treatment. So you're sort of looking at recovery over time. Now, if we focus in on the two solid lines, the blue and the yellow one, they both represent the effects of 20 milligrams THC. It's just that the blue one is inhaled and the yellow one is oral. And what we're interested in is the point at which these lines intersect with the impairment threshold, or which impairment becomes negligible. And as you can see, this seems to occur at about the 5.5-hour sort of time point with inhaled THC and 8-hour time point with orally ingested THC. The dashed lines represent lower doses of THC and therefore correspond to shorter recovery periods. So overall, we concluded that impairment typically lasts less than 8 hours, but does vary depending on the dose consumed and route of administration. Following this, we went on to conduct a really focused systematic review looking at the next day or more than 8-hour effects of THC. And this was largely on the basis that there's interest in this area from, I guess, safety-sensitive industries. So, I guess, construction, transit, things like that. And overall, what we found, I guess really briefly, is that the vast majority of tests conducted, or performance tests conducted more than 8 hours after THC use, didn't demonstrate any impairment, while only a small number did. Also, we found that those that didn't demonstrate impairment tended to use better quality sort of research methods. So we concluded that there was limited evidence to suggest that THC can cause next day impairment. Lastly, we went on to have a look at the relationships between biomarkers of THC use and impairment. So once again, graphed here on our x-axis, we have our measure of impairment. Y-axis. This time on our x-axis, we have our blood THC concentrations. What you can see in the first graph is that there was a significant but very weak relationship between blood THC concentrations and impairment in occasional cannabis users. And on the other side, no relationship whatsoever between blood THC concentrations and impairment in regular cannabis users. In fact, the graph kind of goes the wrong direction than what you'd anticipate. We ran these same analyses looking at a number of other biomarkers, and they produced much the same results across the board. So this is sort of just an example here. And overall, we concluded that these biomarkers are therefore relatively poor indicators of THC-induced impairment. So in conclusion, we found that CBD does not induce intoxication, impair driving performance, or elicit false positive roadside drug tests for THC, and can therefore be used safely by drivers. On the other hand, THC we found could impair driving performance, but that this impairment typically subsides within 8 hours, though this does depend on the dose consumed, route of administration, and although I haven't had time to discuss it today, the frequency with which one uses cannabis. Overall, I guess this means that we do need to be able to identify cannabis-impaired drivers, but our research would suggest that these biomarkers may not be the best way in which to do this, so further research to be done there. Actually, further research, sorry, I forgot I added this slide, further research to be done in a few different areas. So we need studies looking at the effects of medicinal cannabis on driving, studies looking at drug-drug interactions and the consequences for driving there, and also, as has been raised a few times already today, looking for novel indicators of THC-induced impairment. Thank you.
Thanks very much, Danielle. Thank you. Our next speaker is Llew Oli, and Llew is from the ARC Research Hub for Medicinal Agriculture, and he's going to be talking about towards genomics, assisted improvement of glandular trichome productivity in cannabis sativa. Thanks.
Thank you, Veronica. And another topic on trichomes, and I would like to thank Matthew for the nice introduction to the topic. So this, my research project is one of the several research projects that's currently ongoing within the medicinal agriculture hub. And in this specific project, we are using the latest genetics and genomic tools to try to understand trichome development and then enhance trichome productivity. So one interesting thing about cannabis as a plant is that it does produce male and female flowers on a separate plant. So a plant is either male or female. So one striking difference between these two sexes is that the male plants produce more abundant flower buds, and then these flower buds are covered with this hair-like structure, what we refer to as glandular trichomes. So the reason why we are interested in these specialized structures is that this is the site where all the interesting chemicals are synthesized and stored. So traditionally, cannabis breeding, people have always tried to optimize flower production. That means the more flower buds produced, and you get high yields. However, within this research project, we are really trying to look at trichome both as a structure and also what goes within the structure, that's all the biosynthetic pathways. So the key questions we've been trying to answer is, how are these glandular trichomes initiated? Because they are epidermal outgrowths. So how are they initiated? What are the kind of developmental stages they go through to reach the full size where they start synthesizing and storing the chemicals? And also, we really want to try to understand what are the key genes that are involved in this initiation and development. Not only identifying individual genes, we also would like to understand the key players that are the regulators, because sometimes it's really hard to target several genes, but if you can identify key players, we can target that so that we can optimize trichome yield and also productivity. So we know from other model species, like tomato, there are several key players that control both the number and size of trichomes. So you can increase number, you can also increase size of individual trichomes. So we think that we can learn a lot from this existing knowledge, but also try to understand whether similar processes happen in cannabis, whether this process is conserved or we can find unique players within cannabis system. And also, we do know a lot of the genes that function as enzymes in the biosynthetic pathways. But still, we don't really understand how these genes are regulated. So another important component of my research is trying to understand and characterize these enzymes, and also, like I said, identify key players or regulators so that we can target them for improvement. So one of the tools that we have been using in this research is to try to develop a transcriptome atlas, that means RNA sequencing, try to understand what gene is expressed in what tissue of cannabis. So this is an example where we have the flower, the stem, the leaf, and trichomes and roots, and see what genes are specifically expressed in different tissues. Because we know that these specifically expressed tissues are important for tissue function. So if we can identify trichome-specific genes, that could give us a clue of what are these, the function of these processes. So those in the heat map, so those red bands are so the regions that specifically tells you these are the genes that are specifically expressed in different tissues. Like I said, we have identified a lot of the individual genes that are involved in these processes. However, we wanted to go a bit farther. We wanted to really know, understand how these different genes interact, because we know that genes often don't really function in isolation. They usually interact. So this is, there's a computational methods that you can use try to predict the interaction between genes. So this is like a gene regulatory network that tells you that there are genes, these genes interact with this gene, and then it regulates or controls the expression of a downstream gene. So all those circles in colored circles are the potential regulators within the trichome, and the small black dots are the genes they can potentially target. So the long-term goal of this research is to identify these key players and target them for selection and also for targeted modification in trying to improve trichome productivity. So I would like to thank all my colleagues from the Trobe University and also from our main industry partner, CAN groups, who have been involved in this project, and these are all the list of our partners. Thank you.
Thank you very much, Llew. Now for a bit of heresy, which is the production of cannabinoids without using plants. We have Dr. Adam Amatovsky, who's head of MedChem at the Lambert Initiative.
All right. Well, thankfully for you guys, I'm actually not going to really talk too much about actual synthetic chemistry today, so apologies to the very few medicinal chemists who are here and probably actually looking forward to this. It's more of an overview of the medicinal chemistry operations at the Lambert Initiative. So we're lucky enough for the Lambert Initiative to be sort of comprised of a multidisciplinary team, starting from medicinal chemistry, moving through to molecular pharmacology, analytical chemistry, in vivo pharmacology, and then our clinical and policy sort of research teams. And this sort of is a really good opportunity for like rapid translation from discovery in medicinal chemistry all the way through to the clinical labs. So this talk will focus a bit more on medicinal chemistry strategies, glossing over synthetic chemistry, and how these interact with the other teams at the Lambert Initiative. So within the medicinal chemistry program, there's sort of 4 primary research programs. I'll speak about the first 2 today, so they're both sort of related to phytocannabinoids, but suffice to say we also work in the orphan GPCR space, as well as repurposing synthetic cannabinoids, looking at sort of developing new therapeutics for pain and inflammation. So firstly, just covering our program on phytocannabinoids and generation analogues of these classical phytocannabinoid scaffolds, sort of with the aims of generating novel chemical entities, sort of mainly in the space of epilepsy and antimicrobials, that potentially have improved ADME properties and efficacy in these indications. So due to IP sensitivity, I can't really show specific structures today, but I will describe sort of the types of modifications we look into and the scaffolds as well. So within the phytocannabinoid class, we look at sort of all the major phytocannabinoids such as THC, CBD, and CBC, but we also have some interest in the lesser known ones such as CBE and iso-THC. And primarily we look at systematic sort of modification of sort of the tail regions shown in orange here, but we also look at some lesser explored areas shown in blue. So these move into our sort of molecular pharmacology program. And so we run basically a multitude of assays on most analogues. These are sort of high throughput. So on the left, you can see in the wagon wheel, these are the sort of scaffolds that we're able to look at within these particular assays. So we routinely run CB1 and CB2 functional activity on all our molecules. So this is to ensure there are no liabilities with intoxication arising from CB1 activity where we don't want that, but also to discover novel activity at CB2 receptors, sort of which can be desirable in a range of indications, sort of including inflammation and epilepsy. A more recent addition to our program is looking at the antimicrobial activities of these compounds. So this is actually run in collaboration with Mark Blaskovich at UQ. And so highlighted in the bold up there is sort of where we've actually had some hits in these spaces as well. So we've had some exciting results come through within our CBD series of analogues and our CBC series of analogues. So these are just a few examples of what we do in the in vitro pharmacology. So we also look at membrane permeability, but I won't go into that here. So that's more in the synthetic cannabinoid space that we look into that assay. Okay, I think the control might have died. We're getting a manual change over there. All right, great. Oh, got a back one. So yeah, within our in vivo pharmacology program, we have a focus on seizure models. So sort of starting at the top, we look into the MES seizure model in rodents. We've had some interesting results, unsurprisingly within the CBC series, but also looking at these iso-THC scaffolds, which are sort of much lesser known, so that's kind of exciting. Our bread and butter is more the Dravet syndrome model in rodents that we look at. And we've had some hits in CBD and CBC series, and we're hoping soon to get a zebrafish model of Dravet syndrome up and running. So that's sort of in the works, which is much higher throughput, and we should be able to look at a sort of broader variety of our compounds. We do have a working model of inflammation in zebrafish, and whilst we haven't really had any hits in the phytocannabinoid space, a couple of our repurposed synthetic cannabinoids actually have been quite promising in this area. So moving on from the phytocannabinoid analogues or modification, we also look at synthesis of tool phytocannabinoids for use as reference standards, but also to look at their activity in earnest. And this includes metabolites of some of the major phytocannabinoids themselves. So an example of where this applies at Lambert is our interest in cannabinoids and sleep. So anecdotally, you probably know CBN has been referred to as the sleepy cannabinoid, and we have several projects running through all of our teams which sort of aim to clarify this assertion. So to study the effects of drug in vivo, you need access to its metabolites. So the primary metabolites of CBN you can see up here is 7 or 11 hydroxy-CBN, whichever way you sort of want to name that, and the carboxy version as well. So these are needed so you can determine their pharmacokinetic profiles of the parent compound, and of course in a clinical setting analysis of blood samples and the like. So these metabolites of CBN are scarcely available commercially, and this is particularly true of larger scales which are sort of required for preclinical studies, which we're also interested in. So within the medicinal chemistry lab, I sort of developed a new synthesis of both of these metabolites, able to sort of create them up to 500 milligram quantities, which has supported the preclinical rodent work that we've used them for. So this feeds into, preclinical research, analytical chemistry, and clinical research. So we're able to look at, sort of the in vitro pharmacology of this compound, its PK profiles of these compounds, its PK profiles, and of course then this will also allow the analysis of blood samples coming from the Cupid trial, which is looking at the effects of CBN on sleep architecture, which is currently ongoing. So with that, thanks for listening, and I'm happy to take some questions at the end.
Thanks very much, Adam. And next speaker is Martin O'Brien, and Martin's also from the ARC Research Hub for Medicinal Agriculture, and he's going to be talking about the use of tissue culture into a commercial production line, the hows to get there. Thanks, Martin.
Thanks, Veronica. I would like to say it's a great opportunity for me to present our latest successes within the MedAG Hub, and all that research has been conducted within the walls of the research building of Can Group Limited. Is this reliable? We'll get there, but yeah. So yes, I'm working on tissue culture. This is the potential to grow plant in a sterile manner, in a tube, plastic, glass, and with synthetic media. And the media we provide will kind of dictate the shape the plant will have. A lot of meristems, long nodes, quick growth. So yeah, we have a way to modulate how the plant grows. And the tissue culture technology has benefited some industries that use it quite a lot. The forestry industry, the ornamental and flowering plants. So all the cheap orchids you see at Bunnings come from tissue culture. And then there's the banana industry as well. So all those three industries have benefited quite a lot from tissue culture. But you're not here to hear about orchids, eucalypts, and bananas. So let's stay on topic and test this. Haha. Yes, good. Okay, thanks. It was a laser. So here we've got a little pictures, a table from a paper that kind of showed advantage of using tissue culture compared to the traditional ways of having a crop in a medicinal cannabis. And what you can see is there tenfold potential increase in profit. And this is all come to the amount of clones that can be generated in tissue culture per day by a single person, the amount of space those plants takes per meter square compared to the real plant, and the fact that we can work on those plants, step away, and let them go for a month without even any care. So there's a lot of cost reduction that make this profit line. And our aim is to try to help the industry implement tissue culture and reaping those benefits if possible. So first, I would like to show you the traditional way to grow cannabis in the industry. So you've got a mother plant that produces around 50 cuttings each that feeds into a production line, and those will be flowering, and then a product will be taken out of those plants. Then to rejuvenate those mother plants, which needs to be done every 6 months, the cuttings will be used to go up and then create new mothers. What we're suggesting is using tissue culture. And with tissue culture, the arrows can go anyways. We can have the tissue culture producing mother plants. We can have the tissue culture producing actual flowering products. And yes, we're saving space and saving on cost as well. So I'm going to show you 4 examples in the last year where tissue culture has been a benefit for the industry, or what we've been doing over at CANS. So first, when I first started, we've been handling several cultivars, a bit over 20, and some of them were fuzzy heaters. You all have those in a family. And yes, so it was trying to find specific condition to grow them all, and a happy condition for them to be usable for either bring them into a production line or maintaining them constantly. So some of those are 3 years old, they haven't been having to go back to the mother plants. And you can see in the first big one, this is one germplasm or one cultivar that behaves quite goodly in one media. But if you look at the other 3 little pictures, these are other cultivar, and they're not so happy in that media, or they have a different range. So we have to tailor conditions. So we've got now all those lines that are now in tissue culture and can be maintained indefinitely. Another aspect of this thing is the ability to pivot in time of crisis. So very recently, someone from the cultivation team came to me and said, Martin, we've got an issue with one plant. It's virtually dead, or it was nearly there. That's the picture on the left. And asked me if there was a way to rescue this plant. And 3 weeks down the line, through tissue culture, we were able to maintain and save this plant. So there's a way with tissue culture to, if there's a problem in a glass house, contamination, we can bring things back, maintain them in a sterile environment, deflasking them back to the glass house. Now, I think we're going to go on the more important aspect. I've got a minute to do that. One of the possibility of the production line is to bring tissue culture into mother plants. So replacing mother plant in a very high throughput, and once they produce their 50 cutting each, those mother plants are discarded. Having tissue culture feeding over and over some other plants. Then, yes, those mother plant feeds into the flowering product. And to test this, we grow some tissue culture plant side by side with the traditional method, and we test the amount of cutting those plants were able to produce compared to the cuttings. And as you can see, depending on the age of those plants that spend time in tissue culture, that actually they can produce more or approximately the same. So this is a tick that can be, this technique could be used to replace mothers. Another approach would be to use tissue culture into feeding into the production line directly. And to do this again, we grow those plants side by side to the traditional method of cuttings. And we evaluate the productivity at harvest, but also the cannabinoid content after drying the material. And what you can see on the left side is that the harvest index is the same, depending if they come from tissue culture origin or from a cutting origin. And on the right side, you see the amount of THC in those plants, and they exceeding the amount of the cutting method, but I wouldn't bite too much into this. But yes, tissue culture could be used into a production platform. I'm already over time. So I think given the right media and the right cultivars, we can achieve great things in tissue culture, and potentially we can have a shift in the industry. I would like to thank important people that help out during that process and experiment, and our partners. Thank you for listening.
Thank you very much, Martin. That's excellent. And our final speaker in this session is Dr. Anastasia Sureyev, who's going to talk about the emerging role of cannabinoids in sleep.
Okay, thank you, Iain, for the introduction, and thank you for the opportunity to speak today. So I am a research fellow within the clinical team at the Lambert Initiative, and I'm involved in a number of clinical trials across pain, mental health, neurological disorders, and sleep. And today I'd like to focus on sleep, in particular insomnia disorder. So as you know, insomnia disorder is the most prevalent sleep disorder that affects up to 30% of the general population. And despite treatment, 40% of people will experience persistent symptoms. Medicinal cannabis is becoming a very popular alternative to common sleep aids, and sleep disorders are the third most common reason why people use medicinal cannabis in Australia, and this is based on prescribing data through the TGA and through cross-sectional surveys that we run every 2 years that Llew talked about earlier in this session. So I want to talk about two clinical trials that have been completed with insomnia disorder, both THC-containing products. So first, our trial in partnership with the Woolcock Institute, which used a 20-to-1 ratio of CBD to THC in people with chronic insomnia disorder, which showed that a single oral acute dose containing 10 milligrams of THC and 200 milligrams of CBD had no change in subjective sleep quality. It decreased total sleep time on polysomnography, so our objective measure, and it suppressed REM sleep by 8%. Now, a second study completed by the University of Western Australia, which used a similar trial design and similar formulation, administered the same dose of THC but over 2 weeks, showed that there was an improvement in subjective sleep quality, which is not coming up. Apologies. I'm just going to press harder, see if that helps. It's not helping. Do we have anything else that we could use? Or just a... Thank you. Oh. So there was an improvement in subjective sleep quality, which was corroborated on actigraphy but not on polysomnography, and there was a non-significant trend towards a suppression of REM sleep, so suggesting possible tolerance to this effect over time. Now, the difference between the studies may be explained in part due to the moderate initial starting dose used of THC that was administered acutely in participants that were more or less cannabis naive. So it may have inadvertently stimulated them. As we know, cannabis, people who are not regular cannabis consumption, they will have greater sensitivity to cannabinoids. So we purposefully used a moderate dose to see what effects that it had on sleep architecture, and the REM sleep suppression effect is a novel finding and may have clinical implications for other sleep disorders. Now, what about CBD for insomnia? So there are no published studies to date looking at the effects of CBD for insomnia, and CBD does not appear to be sedating, at least in low and moderate doses. Somnolence has been observed in epilepsy trials where high doses of CBD were co-administered with anti-epileptic drugs, resulting in drug-drug interactions and side effects. And also at very high doses of CBD administered in healthy volunteers, and we're talking 1,500 to 6,000 milligrams. Now, unlike many hypnotics such as benzodiazepines and even alcohol, CBD does not disturb sleep architecture in healthy individuals, which is a good thing. CBD may exert effects on sleep via an anti-anxiety mechanism if anxiety stress is driving the sleep disturbance. And in our recent review, which was published by my colleagues and I, we showed that this effect starts to emerge at the 300 milligram or higher dose with CBD. Now, there's also an emerging preclinical evidence base to suggest that CBD may be alerting and may have wake-promoting properties, and this has not yet been explored in humans and certainly is worth further investigation. Now, to bridge this evidence gap, there are now 5 clinical trials looking at low-dose CBD and insomnia, 4 of which are registration trials, and I've just summarized a bit of information about these trials here, and I'll talk you through that. So all of them are using a double-blinded, placebo-controlled parallel trial design. And the inclusion of participants based on their ISI score varied. Due to the waxing and waning nature of insomnia, a high ISI score may provide more room to identify change, given the strong placebo effects inherent to insomnia trials where a subjective outcome is used, such as sleep quality. The treatment period varies, so if we got from 2 weeks all the way up to 10 weeks, the range of doses up to 150 milligrams were used, and the yellow star here indicates formulations with alleged superior absorption of CBD relative to standard products. Most used a change in ISI as the primary outcome, and so far 3 trials have reported, this is just their preliminary top-line finding, is that CBD was not superior to placebo. Although disappointing, we do have now a wealth of data on the effects of low-dose CBD in sleep, which may help us to then inform future research. And certainly looking at secondary outcomes of this trial will be of great interest as well, and potentially there may be a signal there. I just want to talk briefly about other non-intoxicating cannabinoids that are being explored in insomnia. So as Adam mentioned previously, we're also investigating cannabinol or CBN, and the low and high dose that is being administered in the Cupid trial was informed by preclinical research that was conducted by a PhD student at the Lambert Initiative. So we are looking at different cannabinoids in this space. Other trials that are being conducted overseas include CBG and a CBD and terpene combination in insomnia disorder. So I think these are both US trials. So just to conclude, looking at future directions, exploring lower doses of THC that confer clinical efficacy without significantly disturbing sleep architecture is certainly of interest. Exploring THC for REM sleep state disorders, so things such as REM sleep behavior disorder and Parkinson's disease, as well as trauma-related nightmares and PTSD, certainly of interest there. And finally, identifying novel non-intoxicating cannabinoids such as CBN and others are certainly of great interest, and this is something that we're doing currently in our preclinical pipeline, so watch this space. Thank you so much for listening.
Thanks very much, Anastasia. And we have a couple of questions on the Q&A, but is there any questions in the room? You'd like to put your hand up, and we have a roving mic for our panel of presenters to answer. We've got one over here. Yep.
Excellent job, all of you. I learned quite a few things. Just a quick question. For those of you that describe doses of THC or CBD, can you tell me about the vehicle and if it was in fed or fast state?
Okay, sure. So I can only really speak for the trial that I helped coordinate and design. So it was orally administered. The excipient was MCT oil, and so there was no kind of particular technology associated with that particular formulation. And in terms of the dosage, was that the other part? Fed or fasted, yes. So because these were people with insomnia, we did give the drug an hour before bedtime, I think, and they had dinner prior, so they would have had dinner several hours prior to dosing. So it wasn't immediately prior to dosing, but several hours prior.
The meta-analyses I described in the systematic review obviously varied from study to study. The initial study I described looking at the effects of CBD on driving performance, the CBD was given in MCT oil solution alongside breakfast and a high-fat dietary supplement to try to maximize uptake. Thanks.
I think we had a question in the centre here.
Maybe whoever's answering could just jump up to the mic. We've only got one. Unless we've got another roving mic.
Thank you. Really great talks, everyone. I have a question for Martin specifically. Tissue culture. Is it possible to regenerate a plant from callus? And I'm thinking, for example, that's what you use for genetic transformation.
So people have been using embryonic calli to achieve this, so there's evidence for that. But somatic tissue, like a piece of leaf or a piece of stem, petiole, there's no.
Should we go with these?
Before you leave the stage. It's one of the things I think we've all been waiting for is tissue culture to really be able to supply cannabis production. How does using tissue culture affect the architecture of the plant when using it for production?
I'll be brief. It does change the architecture. And I think it's the nature of the plant. It comes out of tissue culture with very short node, and from there you've got a lot of basal major branches. And yeah, I'll leave it to that because we think it might. Is research being done in that space? It's all depend of the. So you saw some of the plant that I've used had from 1 week to 6 weeks. If they've been spending less time in tissue culture, those compressed nodes are less. There's less compressed nodes. If they spend a lot of time, there's a lot of nodes. You've got only 10 centimeter of growth. If they've got 8 nodes compared to the 1 week who's got 2 or 3, the architecture change slightly between those. Yeah.
Question up the back.
My question, sorry, I forgot all your names. The fellow that spoke about the terpenes and the beta-amyloid. I found that really interesting. I didn't realise that terpenes had an effect on the beta-amyloid. So my question, and I'm not a neurologist, I'm just asking the questions. One, do you know the mechanism of action? But also, is there evidence to show that beta-amyloid is actually the cause of Alzheimer's and not just a marker? And if it is the markers you're looking at, have you also considered repeating the study also on tau tangles, which are also a marker of... Yeah. Go for it.
Yes, so it is a hypothesis, the amyloid beta theory. I wouldn't say it's the only cause of Alzheimer's disease or even dementia specifically. And as far as I know, at the same sort of time when the accumulation of amyloid beta is in the extracellular fluid, the tau protein grows intracellular. And I have not come across a model yet in the cells that I'm using that would demonstrate any sort of system to test the tau protein against the terpenes. Molecular mechanisms of action is the next part of the research to figure out how they either are being neuroprotective and some are being neurotoxic as well to the cell line that I'm working with. Did that? Yep, and so some are neuroprotective and some are not showing neuroprotection inside this neuronal cell line. So stay tuned for more molecular mechanisms of action. That's my last year here. So.
And John, while you've got the microphone, there's one on the Q&A for you, and that is, can consumed terpenes maintain sustained levels in the brain such that a therapeutic effect is possible?
More areas of research. I mean, they're highly bioavailable, but they're also excreted within 2 to 4 hours through CO2 conjugants. So basically, you can inhale them and you can exhale them almost just as quickly. So what sustained levels are they? I mean, aromatherapy might be a tool for that, where it's constantly infused into the air. But so far, what I've read so far, it does penetrate the blood-brain barrier, so there is potential there. But if anybody has ideas of how they measure that inside the blood-brain barrier, I haven't come across that pharmacokinetic area yet.
We also had a question on the chat from Donna Edmond asking why use zebrafish. Maybe Adam would like to answer that.
So this isn't exactly my forte, but I mean, as I sort of touched on, it's able to be done in sort of quite a high throughput manner, and it is largely, a lot of assays with zebrafish are able to be done with automation as well, with like cameras that can do tracking and these sorts of things. Compared to rodents. I think if I'm not mistaken, the husbandry time, so like growing them as well, is actually quite a bit shorter. And you can do CRISPR on these as well, which gives you like a good range of assays that you're actually able to look at. I don't know if anyone has anything else to say on that.
Yeah, I think extremely high throughput is probably the three words you would use compared to rats and mice and other mammalian models.
I think we've got a question coming.
Have we got any more questions? No? Okay, well, what an excellent session, and the future of cannabinoid science is in very safe hands with such brilliant researchers, and they all kept to time as well. Amazing. Thanks, guys.